Thank you all for coming to the seminar today. It's my pleasure to introduce Dr. Brian Barnes, who does prefer just Brian. He is a research assistant at Florida, and his interests include improving satellite materials, calibration and validation of ocean color satellite data, assessing continuity of satellites for ocean color research, and he is a member of the NASA Science and Application Team and the International Working Group focused on Sargassum, satellite calibration, benthic reflectance, and satellite systems. He holds a master's degrees from the Virginia Science College, Mary, and a PhD.
Thank you so much. Cool. Thank you so much. Thank you, Jen, for inviting me to come speak to everyone today. I hope everyone can hear me online, everything. I think we did the sound check, so we're just going to charge right in. I will start by, of course, acknowledging I'm going to present a lot of research here, and I am not the one that did almost all of it. So most of the people, the collaborators that I've listed here, and many other people have contributed to the work and are really who I have to thank for doing this research. And then, of course, our major funding for the projects that I'll talk about comes from NASA primarily, but also EPA and NOAA. So again, without their support, all right, so I understand this is a pretty, the group has a pretty broad background. So I'm going to start with a broad knowledge base here. So I'm going to start with the background of like what I do, what is satellite ocean color as a field. And then a couple of examples, I think, of why it's necessary. And then the bulk of the talk is just case studies for how we use satellite data to study interesting things that are happening in Florida coastal waters. So we'll talk about Carenia brevis, Synechococcus, and Sargassum. You'll see the colors, red, yellow, and orange will change as we move through these various topics. And finally, I have a very quick wrap up, and then it's time for a happy hour. All right, so ocean color remote sensing. This is the cartoon version of what you're going to see from a ocean color satellite. You have the satellite on the right there, and you have sunlight that's coming in. It can it comes from the sun. It can interact with the air and be bounced into the satellite field of view. It can bounce off the surface of the water and into the satellite field of view, or it can take the long path into the water, get bounced off by something in the water, and back out and be measured by the satellite. And all we are doing with ocean color remote sensing is trying to use that part of the signal to identify what's in the water. So if you have something like some diatoms here in the water, they're going to change the light field that's coming out of the water relative to a pure ocean signal. And you can assess or make some estimates of what is in the water based on the color, the light field that's coming out of the water. There's a further complication in areas that are super shallow and super clear, where you also have to deal with the bottom. And this can change, sometimes sandy bottom, sometimes seagrass, and so forth. So that also changes the light field. And decoupling all of that is quite a challenge. I won't talk too much about optically shallow waters here, but I know some people in the college are interested. It's a key, difficult property to deal with, especially in these coastal areas. The other thing is you will see in a lot of the maps that I provide just kind of either black or gray or white or something like that, some color that's there's no data. And no data usually it's because they're clouds. Ocean color remote sensing does not work in the presence of clouds because the light can't get to the satellite, can't see it, and so forth. So just be forewarned that's why. All right, so I talked about the color of the water. What do I mean by that? So a satellite or any kind of radiometer will measure something like this. And the x-axis here is the wavelength of light. And on the top, I've got the rainbow that corresponds to those particular colors. So we're measuring in the green and in the blue, separate from in the red, all together. And based on the color, again, of a water parcel, you can identify what's in it. If it's super bright and turbid, it would have a spectral shape that would be pretty flat, but very reflective. So it'd be high reflectance. And reflectance is the term on the y-axis. Looking at chlorophyll specifically, though, if you were talking about a parcel of water that's pretty low chlorophyll, it's this blue, the green line here. The green line it's reflecting more, you get higher reflectance in the blue spectra, the end of the spectra, and then it fades away as you get into the red. That's why the water is blue. But as you get chlorophyll in there, and if you look at that blue line, as you start to increase your chlorophyll concentration, the chlorophyll is absorbing that blue, and that's why you have low signal here in the blue. And then you have a little bit elevated in the green. The chlorophyll is green, it's scattering in the green, and that's why you see that signal. As you get more and more chlorophyll in the water, your spectral signature starts to look a little bit different. And I just want to highlight a couple of features here. I don't know if you guys online, whatever. Okay, so these features here, this one, these are fluorescence signals. So essentially this, as chlorophyll is absorbing the red light, it kind of has an inelastic scattering, and it fluoresces in a red wavelength. So that's something that'll come into play in a little bit, this fluorescence signal. But in general, this is kind of what I do, the nuts and bolts behind how it works. And I'll highlight one other point is that not all chlorophyll is built the same. Not everything has the same spectral signature. So you might have a shoulder or something in the data right here that is particular to a specific chlorophyll pigment or something like that. So that'll become important later. All right, so why use satellites? We've got a platform that's 700 kilometers above the Earth, and we're looking at an area that actually has been pretty well studied. Many people in this room and so forth have collected in situ data in the area I'm picking on here is the Florida Keys and the Florida Bay area. Tons of data that have been collected here. Why do we even need satellites? And I don't want to disparage anybody's work that does in situ data collection. It's valuable and it's critically important. But satellites, they pass over every day, and they're collecting data that are, covering, with the exception of clouds, covering the whole swath of this area. So if we can tie what the satellite sees to those data points and make sure that we've got a signal that's consistent, then we can kind of fill in those gaps in both space and time for using the satellite data set. And while we are doing our research as far as using satellite data to identify water quality issues and study water quality in coastal environments, one thing that there's a pretty high barrier to entry for things like satellite ocean color data, and getting from what the satellite sees to something that's usable to a resource manager is a tall order. So a big part of what we do is not just derive products and do science on converting what the satellite sees to something real, but we provide those data online. That's almost all of our projects have a system whereby we provide the data to end users. And this is essentially what that looks like. So we have a satellite that's passing overhead, it's looking down, taking measurements. It's sending those measurements to any of their, whatever space agency launched the satellite. So this would be NASA, ESA, and so forth. And those agencies ingest the data and they post it online and we grab it. We grab it in near real time. This is all done autonomously. So within a couple hours of overpass, we will have data in our hands. And we take those data, we process them to derive things that are important to researchers or to resource managers or whoever is interested in them. And we shunt it to our web-based data distribution system. This is all done, it's publicly available, it's all done in near real time. And this is kind of the core framework for how we provide data. And an example of such data would be this. This is a true color image collected by the Sentinel-3A sensor. This was Saturday. And I love this picture because it's got a nice clear view of most of Florida, which you don't always get. And also there's a lot of really cool stuff happening in the water around Florida right now. So starting from the beginning, let's zoom in on Tampa Bay region. This is where I come from. We currently have a red tide going on in Tampa Bay. Red tide is caused in this area by Karenia brevis, which is a dinoflagellate. It looks like this in SEM. It's and there it is also on light microscope on the right. And if you haven't experienced a red tide around Florida, you don't generally get them over on this side of the state that much, but it's associated with fish kills, and you have you don't want to be eating any seafood or anything that's been impacted by the red tide, and also it creates a brevetoxin. That's what causes the fish kill, but it also causes respiratory distress. It's really nasty for some people. So they will do beach closures and so forth when they have a persistent or a sufficiently sized red tide. And we can see red tide from the satellite. But just like I mentioned when I was showing the spectra, the different concentrations of chlorophyll, not all chlorophyll is the same. So here I'm showing just the red here is higher chlorophyll, the blue and purple is lower chlorophyll. And you see a pretty common gradient that you see offshore, clear water. As you get onshore, you start to have higher chlorophyll. And that's all well and good. We can say, well, there's red tide near shore. Well, that's not really true. I'm overlaying now some samples that were collected by the Florida Fish and Wildlife. They go out and they do regular red tide monitoring in situ. They go out, take a sample, put it under a microscope, and you know for sure whether or not red tide is there based on that method. And if you're matching together what the in situ data is showing and what the satellite data is showing, let's look here, especially in Charlotte Harbor area, you've got huge amounts of chlorophyll, but no red tide. Okay, so clearly not all red tide or not all high chlorophyll is red tide. So we have different algorithms, different ways to interpret the satellite data that look. I mentioned that fluorescence signal. Now the red tide in Florida, it fluoresces much more strongly than others. So we use that as one of the ways that we can identify Sargassum, identify red tide. And you see a lot better coordination between what the in situ data are showing versus where we are identifying red tide in the satellite data. And again, we can fill in gaps where they don't necessarily have that action. So these are collected over 7 days around it. So things may have moved a little bit. But this, we can identify that this is a very likely high concentration of red tide, that they didn't go sample. So they can go sample. Additionally, we can overlay this with other models that are identifying prevailing currents. So at this particular time, there was a primarily offshore outgoing current. And thus we can make some inferences that these red tide, even if they're near shore, are likely not going to be impacting beach environments. Put in another way, you can do each individual point as a tracer, and you can follow the course of this individual red tide particle through time and see it where is it expected to go over the next 7 days or so. Right, so that's what we have. We call this our Integrated Red Tide Information System, or IRIS. We're all about acronyms, of course. But where are we going in the future, and what are some of the key things that are standing in the way of continuing this type of work? So the problem is, I mentioned how important fluorescence is to identifying red tide and distinguishing it from other sources of chlorophyll concentration. Not all sensors measure fluorescence, and most important, we have one of the workhorses over the last 20 years has been a sensor called MODIS. Well, it's aging out. It's 20 years old. And what has been replaced with new sensors that do not measure fluorescence. Bad call, in my opinion, but I don't make the decisions here. So even without the fluorescence signal, we are kind of left to use different sources of information. And in particular, in this case, we use artificial intelligence to be able to identify red tide. In this image, you see this is all red tide identified here. And this is that's another source of high chlorophyll water that we don't capture. And it's based purely on the spectra that aren't looking at that fluorescence region. The other thing that we're doing is looking more at coastal areas. For these coarse resolution sensors, I mentioned MODIS, it has a resolution of 1 kilometer. Okay? So, and even within that, you're wanting to avoid land because your uncertainties are super high in land. So you take even a step farther away. You're at least 1 kilometer away from land before you even start thinking about processing data. Well, I know many in this college are using SuperDove or the Planet data set. And here's an example of how we can use it to visualize. You can't really classify, you can't really get into quantifying red tide using these systems yet. But nevertheless, you can visualize red tide inundation at a scale that is just completely unimaginable using these other sensors. And I want to highlight too, like how spatially dynamic these systems are. You think of red tide and you think of just like, oh, it's a big red blob. We'll get into blobs later, but it's not. It's got these feathering. It's got all of the hydrodynamics come into play, and it goes into these small little embayments with the prevailing water motion, and we can capture this using the system. I'm going to drive that spatial scale point home one more time. This is the same exact image where we're zoomed into a really fine scale, region here, and you see these filaments of red tide that are impacting various areas. This is a kind of high resolution sensor, but not that high. You can kind of maybe see stuff. And this is our traditional data set here. You know, where this is the white is the 1 kilometer from the coast just to start. And then you've got these black areas where you just don't have good data, you don't trust it. And you're really grasping at straws if you're trying to infer anything important about these coastal areas from those course regulations. So that's how we move from being an offshore-based warning system, as you will, to being something that's really measuring at the level of beach, which is what people are interested in. Care if red tide is 2 kilometers, 3 kilometers offshore, as much. It's more so when it's on the beach and affecting your particular environment. All right, so wrapping up red tide, so that fluorescence signal is critical, and as we're migrating to sensors that do not have that signal, we're having to use new approaches like AI and so forth. And we have developed a system that uses satellite data. It incorporates in situ data in near real time. It incorporates those coastal modeling, so the current vectors and so forth. All of that is combined in near real time and presented through our website, through that IRIS system. And then, going forward using high resolution sensors, that's really how we're going to advance the capacity for satellites and red tide monitoring moving forward. All right, so let's back out of the Tampa Bay region and let's go now to Florida Keys region. And here we're talking about a different critter, and this is Synechococcus, which is a pico nanoplankton. It's a micro cyanobacteria, and it creates blooms in Florida Bay and associated areas. And we care about it because when it creates those blooms, it can have pretty nasty impacts to things like sponges that you got here. This is a sponge that's in pretty rough shape due to the yellow water above it. And then also seagrass meadows. This is similarly type impacted, a system impacted by a Synechococcus bloom. And as I said, there's a lot of cool stuff happening. They're in the, coming here down this way, you can see kind of a pea yellow color to some of the water. That is the bloom. You can see it from satellites. And we have a cool method to use a specific spectral feature of this particular specific cyanobacteria to identify chlorophyll that is associated with this specific class. So just like I showed you before, if we were to look at a chlorophyll map, the chlorophyll here is high everywhere, pretty much. But these areas are using that specific feature of one of the pigments in this critter. We can identify which blooms are a part of that as opposed to just in the background level. Taking it one step further, you can use this to watch the progression, literally watch what's happening over time for a particular bloom. And this is essentially daily imagery. You'll see it accumulating. There are a couple of gaps in there that you'll know when you see them. But more or less, the blooms will start in oh yeah, there was a long gap there. That's why the blooms spread out. The blooms will start kind of in this North Bay area up here. And they'll kind of spread out, propagate, they'll elongate, and then eventually they'll peter out. All these images are collected over about a 3-month period. So looking at the same data just in another way, we're looking at what is the extent of the bloom. And we see over the last dozen years, we have more or less 2 cases. You've got some years where you're pretty much flat over the course of the year, and then other years where you see a Synechococcus bloom that happens in the time frame of September and then termination somewhere in the January time frame, early January. We call it normal, although there's really no normal in Florida. But there's also huge deviations. If you look at water year 2023 here, started way early. It lasted for almost the entirety of the year and so forth. So these are the type of events that we want to study. Why is that year different? And why do we see differences in the timing, the duration, and the intensity of each of these things? Looking at the location for where these things happen. So I mentioned that we usually start up here and then we kind of fan out. Some years you get this type of impact where you're going through the canals and impacting the ocean side of the Florida Keys. And this is where you have reefs and seagrass beds, of course. There's also on the very side. But That's another key impact, that these blooms, they're coming all the way from the Everglades, flushing all the way out through the key systems. But some years it looks a little bit different. This year particularly, you have it hanging out mostly in the northern portion. And this year it kind of was a little stunted growth. It was a much smaller bloom. So this is the type of aggregated analysis that we do to identify both when and where these are happening. And then cutting it one step further is the why. And to do that, we are looking at where these blooms come from. So I highlighted that north central Florida Bay region. In earlier, this is kind of where we first see the bloom, somewhere in this region. And I'm going to highlight there. So there's a bunch of lake systems here, and I'll mostly be focusing on these. But these are called, all these are called the I-calls, or at least I don't know how general that term is, but it's the intermittent closing and opening lakes and lagoons. And these I-calls for a long time by many other people than me have been proposed as either providing the seeds for the cyanobacteria blooms, or they're providing nutrients that support the cyanobacteria blooms, or both. So we want to see if we can see that. And to do so, we are using, again, I keep coming back to high resolution imagery. So we're using high res satellites. This is the Sentinel-2 data. And we can see coming from this here, with relation to providing nutrients or organic material that can feed blooms, you can literally see it. You see a pulse of organic matter coming into Garfield Bight here. So yes, you can see it. And associated, this is one of those coarse resolution sensors. You can see this is Garfield Bight here, matching this. You can see at the same time as we had that pulse of nutrients or a pulse of organic matter, you also have a pretty strong cyanobacteria bloom co-occurring with that event. So not at all causative, but it's good to see these things happening at the same time. We also have collaborators who have collected data in the region. So this is another. This is Rankin Bight here. So before we're looking at Garfield, this is Rankin. So collecting data here. This is measuring the chlorophyll. We want to know, like, we're deriving cyanobacteria concentration, chlorophyll concentration in these waters, but is it real? Well, looking at this, the answer is yeah, pretty much. The in situ data here are in the green, and all the satellite data are in the black. Yeah, there's some hits and misses, but overall we're capturing the larger trends. And again, this is where satellites can come in because they provide so many more data points than can be done just with in situ. It's really, really hard to collect a bunch of in situ data. All right, so let's go even farther upstream. So I've got an example here. This isn't in situ data. This is all derived from the satellite. And for this particular year, this is 2020, water year 2020, whatever. You essentially have a bloom initiation somewhere here in like really early July, late June, really early July. And you see this is our chlorophyll is starting to increase in this North Central Bay region, and we see that manifest throughout the Florida Bay. So what about in some of these lakes? We can actually, with some higher resolution sensors, we can look. And in this one of the lakes that feeds this area, you have a pretty high concentration of nutrient or of chlorophyll here, and it seems to die off in October-ish. And then another one, West Lake, just over here, you see a pretty substantial rise in chlorophyll concentration, and then a decrease somewhere in the July timeframe, lining up a little bit with when we saw the initiation in North Central Florida. And I will highlight, again, this is just conjecture at this point, but we had, there was a flushing event that occurred right around the same time, that end of June timeframe. And if you're looking at the salinity in this area, the salinity dropped 10 PSU, which means a lot of water was rushing in there. And right around the same time, you have the bay, or the lakes seem to be decreasing their concentration, and the Florida Bay proper is starting to see increasing concentration. So this is how we'll investigate why and how these individual blooms are being initiated. All right, so wrapping all that up, that phycocyanin, that specific pigment, is what we use to identify Synechococcus, and we have a lot of data that we provide online, daily maps from a bunch of different sensors in order to track this. And we are, where we're going with this is trying to treat this into those, the initiation of these events into the ICALs, those lake systems that are north of Florida. All right, so we'll go away from the Keys and we're going to zoom out even farther to the overall Atlantic. So this is something that I've spent a lot of time recently, which is Sargassum. So Sargassum is a natural habitat. Allegedly, Christopher Columbus wrote about it in his journals. The Sargasso Sea is named for this species of macroalgae. There are two species that live in the Sargasso Sea. And they are more or less like an oasis of structure in the middle of the vast ocean. So you have these floating patches of sargassum. They look like this. They're teeming with life, invertebrates, reptiles, fish, you name it. Everything is living there, being supported by either the structure or the food source embedded within this sargassum. So a hugely beneficial environment. But if you look in recent years, we've seen a pretty dramatic change. Starting in 2011, we have generated, it has been generated, not we did it. But there is now a Great Atlantic Sargassum Belt in a completely different area than we used to see sargassum. And it's having pretty nasty consequences up and down the islands that are, and areas that are impacted. So if you see it in the news, you usually see a picture like this, where just people are struggling to maintain and to deal with a whole bunch of data, or a whole bunch of sargassum. So I often get asked the question, given this, is sargassum good or bad? And the answer is clearly yes. So offshore, offshore, it's actually classified as essential fish habitat. So you can't even take it. You can't remove it without some special permitting, which makes it hard if you're trying to protect your particular area. And then I mentioned the sea turtles. There's 3 species that live in and amongst it that are on the Endangered Species Act. So critical for that. The problem comes, communities there. The problem comes when you get more near shore. And the first impact you have doesn't even have to have physical touching or anything of the substrate. You just have shading, and you can see kind of look at how dark it is under this little patch of sargassum. Anything that's living on the bottom that relies on light is not going to like being shaded out for days or weeks on end. Then you get into other conditions once it's all washed on shore or in an area where it's kind of more stagnant and piling up. It can smother things like seagrasses and corals that are living on the benthic environment. And it gets, often we see pictures related to beaches, but mangroves, this gets fixed inside their interstitial space. And it's incredibly difficult and incredibly devastating to the mangroves. Things that live in it, they can get entrained up in this piling up sargassum and not be able to get out. And then of course there's sea turtles, which they move up onto the beach to lay eggs. There are issues with sea turtle with their reproduction related to the presence of sargassum on beaches. And then one more just piling on effects here is sargassum brown tide. As you got so much organic material just sitting there decaying, you get essentially eutrophication. So you have decreasing oxygen. So this is anoxia, hypoxia, or even anoxia, decreasing pH, nutrients and turbidity increase. This is pretty much standard what you would expect when you make tea. There's also the effects to economies. Of course, tourists don't like piled up sargassum, and it also smells. It's similar to red tide, but without the brevetoxin. It's just hydrogen sulfide. So it's rotten eggs, but it's awful. It's truly awful. And some people get respiratory distress from this. And then finally, there's arsenic, heavy metals, and perhaps even Vibrio in the tissues that are concerns for when you're dealing with it. And this is what it looks like. You'll have a patch of sargassum here decaying, and you've got this plume of water that's emanating from that. All right, so here's a video showing a pretty severe inundation. And just imagine being in this area, either on the benthos as a critter living down there, or even if you're trying to drive your boat, you can't even make it through the absolute incredible thickness of an inundation. Okay, so one thing as we're getting close to the end here, I want to highlight, look at this bay right back here. And notice there's no sargassum there. That's gonna come to a point in just a minute, but I want to highlight that even in this video where you see this is probably the worst thing I've ever seen with relation to sargassum, and even just a little bit down the way, nothing. All right, so what is remediation? What does mitigation look like? So the picture I showed before of people manually cleaning it up is pretty much industry standard now. It's not great. And often that involves also heavy machinery, and that also creates problems because you physically lift up your beach and take it away to a dump site, which, you know, that's coastal erosion, that has huge problems down the road, and you're doing this multiple times a season. If you've got the money, you can install a boom or something like this, and then you can go collect it offshore. That's a great option if you can, although again, there's permitting issues associated with that. And then the other trick that people do is they align it in a manner such that you kind of shunt it onshore, and then it makes it easier to collect. But you also then are dealing with the, you know, you can see the brown water here. You're still dealing with the brown tide, even in the best case scenario. All right, so how do we do sargassum monitoring from satellites? So I showed you the picture of chlorophyll earlier. This is essentially the same exact thing. We are showing different concentrations of sargassum here, and this is the light reflected by them. And the biggest signal that you can see is right here. This is called the red edge, the vegetative red edge. So plants, all plants have this signal here, where you have an increase right here in the near infrared. So higher red edge, we call this AFA, which you don't need to know what it stands for, but higher red edge means more sargassum, more or less. And the less being there are other things that have high AFA. So one is nearshore water here. Just as you get nearshore, you get higher AFA signal. It's just how it works. Also cloud shadows in your satellite image, you will see higher AFA. So in these satellite images, you see not just that sargassum has a high AFA, but everything else. So what do we do for this? Well, here is sargassum, and sargassum kind of has a characteristic look about it from a satellite sensor. It aligns in these kind of long streaks, and it makes these kind of like tear-shaped balls and things like that. So we use AI to look at not just the spectra, so the light that is measured by the sensor, but also the shape, so how it manifests in physical space in order to do remote sensing detection. And with that is how you can, how we build what we call the Sargassum watch system. So the animation you're seeing here is the Great Atlantic Sargassum Belt over the last year. And you can see in October, it starts in this area off Africa. As time goes on, you get into spring, it's migrating westward and getting more broader. It's using up nutrients, and it likes the light and temperature regime at that point. And then as you get into, often we do get it coming up into the Gulf of Mexico and around the Florida Keys. We did not see that this year, not complaining. But overall, this, as you get into September, it starts to die back, and you start again in that kind of just offshore Africa. So now the loop is starting again. So this is the progression of the Sargassum Belt. And I use this particular animation because this is, each one of these is the base map for our data distribution system. So if I were to click on this box, it'll take me to every image that we've collected in the Eastern Caribbean. And you can use that to look more closely at an individual day or something like that. So this is a part of our data distribution that's happening in near real time. We also make what we call Sargassum Watch Bulletin, which is at the end of every month, we take a look at the sargassum in the Atlantic, and we say, here's where sargassum is, here's how much of it, and so forth. And we make some broad scale projections about where it's gonna go and when. So we might say the Florida Keys may see some inundations over the next 2 months, something like that. That's the level of specificity we can get with these forest regulation centers. And it was a prediction somewhat like that got me into, I don't want to say hot water. It garnered some national and international attention, and for about 3 weeks, my life became the man. And I don't mind doing that. I don't love it either. But I especially didn't like when a certain narrative started to take hold, which was that of the blob. Okay, so the blob was coming to take over Florida. It was going to swallow us from Africa. You could walk from Florida to Africa without getting your feet wet on sargassum. I did not.
Professor Schauman, who is one of the leading experts on what many have referred to as a massive blob of seaweed heading to Florida's coast. Fair to call it a blob?
No.
No, we can't call it a blob. Okay.
I would never call that a blob.
Okay, why?
Because it's not.
So that's my colleague, that's Schwamin, who he says it's not a blob. I would not call it a blob because it's not. And he does go on to explain why it's not a blob. But that's cutting it there. But I will note that was a recorded interview. So that means somebody added the chyron later that said, giant blob of seaweed coming, okay? I don't often join in when people call CNN fake news, but you're asking for it at this point, okay? You got your, anyway. Okay, so the reason why this matters is, as I mentioned earlier when I showed the video in the Cayman Islands, Sargassum is super duper patchy. It's patchy out at shore or out at sea. It's patchy when it comes onto shore. And you can have impacts in one area that don't impact another. And the other thing, if you were paying attention to the color scale earlier, the top of this color scale is 0.4%, which means 99.6% not Sargassum. So if you're trying to walk on that red area in our maps, you're in trouble. So nevertheless, I did highlight that from those coarse resolution data sets, we make these monthly projections that are really regionally providing information, but not really helpful if you are on a particular beach. Again, we need to know about beach level because that's where the impacts are being felt. And I'm going to do a little bit of a demonstration here. This is Puerto Rico here in the southwest corner. And this is one of those coarse resolution sensors. And Sargassum here will pop out as green, as will land. So that in the top right, that's land. And Sargassum is here. And I want to highlight, I mentioned the difficulties near land. And pretty much from these coarse resolution sensors, we have to exclude anything within 10 kilometers of land. So we're already looking pretty rough. And then you compare that to what is available using higher resolution data sets. And you really are left wanting for this. So now we can see with much finer detail, we see individual patches, and these are huge. That's half a square kilometer, and that one on the top right. That's a ton of Sargassum. And then each one of those little individual dots is somewhere around the size of a football field. So this is a ton of biomass out there, but it matters what is going to just safely drift off to the next island versus what's going to impact your area. And to bring back the coarse resolution picture, let's zoom in to an area that's really important. This is La Parguera National Marine Sanctuary. And there's the coarse resolution, the high resolution. This is obviously what we need to be using to identify Sargassum and be able to predict with some level of accuracy its impacts. Okay, so we have, just like we did with the coarse resolution, we use artificial intelligence to translate the high resolution images into data products. So I'm going to highlight here, this is the coarse resolution data here. And if you can see the black hard on the screen, that's the 10 kilometer exclusion zone. And then here's the same data collected with higher resolution satellites on the same day. We've actually descaled it by about 50 times in order for you to even see it on this map, because these are tiny patches that are visualized. But offshore, we see the same things. But onshore, these are the areas where you're going to have those impacts. Same thing, this is the same day. Offshore, we're here, and then look at all of this that's completely missed with that coarse resolution. But that's where we're going and in the future of Sargassum monitoring. I mentioned Planet before. We also use Planet data for Sargassum ID and Sargassum monitoring. This is in Mexico. We've got essentially, because of the repeat frequency of this Planet data set, it's high resolution, 4-ish meters. So it's a great resource for this type of analysis where you're watching a highly dynamic system. You see how the Sargassum moves. This is 4 images over the course of 7 days. You can see the difference in the Sargassum location and so forth over the course of this time series. We really need to have this high resolution, high spatial frequency maps in order to effectively monitor Sargassum. And then the next step is coupling it with hydrodynamic models. So this is essentially an image composite. This is us, we seed each pixel is seeding a particle that tracks the location of the water, how it's moving over time. So the reds here are higher concentrations of Sargassum. This is again Puerto Rico. And while this is fine, if you pay attention, again, we have the same problem in the coastal region. This isn't just that our detections are rough in the nearshore regions. our coastal models don't work well in the nearshore regions. You have to have very fine scale models to be able to predict current flows close to shore. And so we are doing exactly that with, again, these higher resolution detections of sargassum and coupled with higher resolution models. And that's how you can, by coupling those, you can make some level of prediction for forecasts. We're also doing this in the Florida Keys, a very similar thing. Each of those dots is an identified sargassum pixel or patch, and we can propagate that through time using the coastal hydrodynamic models that others have developed. I don't do that model. Okay. So let's wrap up with sargassum. It is both an essential fish habitat and a coastal menace. It has a nasty duality, and dealing with it is problematic. We have an existing SAWS program, that's that Sargassum Watch System, that it gives real-time observations. It's used widely for to direct research efforts, to direct remediation efforts, and so forth. And as we move forward, we are working to integrate these higher resolution data products, higher resolution coastal hydrodynamic models, such that we can make forecasts, inundation forecasts, and really have some management links. Instead of saying, hey, the Florida Keys may get an inundation in 2 weeks, we can say this particular beach is at threat within a couple of days due to this particular. So that's our goal where we're moving towards with some of our funding in that realm. And I will close by saying there are blobs in the ocean. That is a blobfish, but this here, the sargassum, that is not a blob. I'll wrap up this. I'm not going to read all this, but this, if you're interested, this is hopefully everything I said about Karenia and Synechococcus and Sargassum. And I'm really interested to hear any questions. Thank you.